So I'm going to go ahead and introduce our next speaker from session 2. Nick Simpson from Iowa State University is an MS graduate student in the Department of Natural Resource Ecology and Management. He has a BS in Fisheries and Aquatic Science from Purdue University, and he worked as a fisheries technician in Indiana and Nebraska prior to coming to Iowa. And his talk is titled Occurrence, Abundance, and Habitat Use of Topeka Shiners in Restored and Unrestored Oxbows in Iowa and Minnesota.
Thanks, Clay. So this presentation covers the portion of my master's project that involves Oxbows. I also do some stream work as well, but this is just, since it's the Oxbow session, just focused on Oxbows. So we heard some of these introductory things in the last couple of presentations, but I'm going to go over them again. So the landscapes of Iowa and Minnesota have changed over time, mostly shifting towards agriculture, and going along with the shift towards agriculture is the channelization of streams, where they're straightened and the banks are very steep. So here's an example of channelization. Looks like my picture is not 100% there for some reason, but you see that the picture on the left has all the natural meanders, and the picture on the right, 150 or plus years later, is very straight. So channelization has several effects in the stream, but for this presentation we're mostly interested in this last point, where channelization reduces the rate of natural oxbow formation, since many of those natural meanders are essentially cut off through channelization. There's less opportunity. What's going on here? And because of the lower reduced rate in natural oxbow formation, there's the oxbow restorations that was touched on in the last presentation, where the sediment is dredged out until the stream bed is reached. So when we start doing these restorations, we end up with two oxbow types. We have unrestored oxbows and restored oxbows, where the unrestored oxbows haven't had that sediment removed. And over time, through flooding events, they fill in with sediment, and the restored oxbows they have gone through that process of having sediment removed, so they're generally deeper and they're more likely to not freeze completely in the winter or dry in the summer. And one of the main reasons for oxbow restorations is the Topeka shiner. So a lot of this was covered before, but it's a native minnow to Iowa, Minnesota, and neighboring states, added to the federally endangered species list in 1998 when they were estimated to occupy only 20% of their native range. Their decrease is due to several reasons, including the decrease in availability and sustainability of off-channel habitats. So when we're talking about oxbow restoration for Topeka shiner conservation, there's some questions that we need to ask, like, are the restorations working? Are they more useful than the naturally occurring unrestored oxbows? And are there any ways that we can update the way that we restore oxbows to increase the chance of Topeka shiner use? So are there any kind of habitat characteristics that Topeka shiners prefer in these oxbows? To start trying to answer some of these questions, I have two project objectives. So assess the occurrence and abundance of Topeka shiners in restored and unrestored oxbows, and to evaluate habitat and fish community characteristics of restored and unrestored oxbows, and determine associations of those characteristics with Topeka shiner presence. So our field work was done in 4 watersheds in Iowa and Minnesota, 3 Huck 8 watersheds of the Boone and North Raccoon and the Rock, and 1 Huck 10 watershed, Beaver Creek in southwest Minnesota. Excuse me. To sample oxbows for fish, we used large bag seines, 1/4-inch mesh, and when we completed the pass through the entire wetted area of the oxbow, we would identify and count all the species that we caught. And along with fish surveys, we did habitat assessments, where first we took water quality. That included temperature, dissolved oxygen, connectivity, pH, and turbidity. And then the rest of our measurements were taken at 3 transects that were equally spaced throughout the oxbows, where we took measurements such as width, depth, recorded substrate types, and other things like canopy cover and in-stream fish habitat. As far as data analyses go, I calculated relative abundance, or CPUE, of each fish species at each Oxbow as the number of individuals per 100 meters squared of sampled area. And then I used random forest tests or models to rank the importance of variables in predicting Topeka shiner presence, and took those most important variables to predict Topeka shiner presence and ran ANOVAs on the log transform data to test whether or not there are any differences in the means for four groups where we have restored and unrestored Oxbows where Topeka shiners were present and absent. So to start with some results, there's a sampling distribution over 2 field seasons. So 98 Oxbows were sampled, 64 of them were restored Oxbows, 34 of them were unrestored Oxbows. The most Oxbows were sampled in the North Raccoon and the Boone River watersheds. And out of those same 98 Oxbows, 40 of them had Topeka shiners in them. And the highest occurrence of Topeka shiners was in the Beaver Creek and the Rock watersheds. And if we look at restored versus unrestored Oxbows, 45% of restored Oxbows had Topeka shiners compared to 32% of unrestored Oxbows. And when we look at Topeka shiner abundance, overall 2,930 Topeka shiners were sampled in Oxbows. 2,500 of those were in restored Oxbows compared to 385 in unrestored Oxbows. And then the figure there on the left shows the average Topeka shiner catch per unit effort in unrestored and restored Oxbows. And if we run a t-test there, restored Oxbows did have a greater average Topeka shiner CPUE. So I'm going to move on to some of the habitat associations or fish community associations of Topeka shiners. So when I run the random forest model, it says that these 9 variables are the most important in predicting Topeka shiner presence. So I'm going to go through each one and show the associations with Topeka shiners if there are any. So the first one is fathead minnow. The figure on the right shows box plots for restored and unrestored Oxbows separated by the line in the middle. And then there's a box for Topeka shiners absent, Topeka shiners present. So for this figure in particular, you see that in both restored and unrestored Oxbows, the Oxbows that did have Topeka shiners present had higher average abundance of fathead minnows. So I put the asterisks on either side of the figure, and in the table we see that fathead minnow CPUE had a positive association with Topeka shiners in both types, restored and unrestored Oxbows. Our next variable is Brassy minnow CPUE. We only see a positive association of Brassy minnow CPUE with Topeka shiners in unrestored Oxbows only. And our next variable was orange-spotted sunfish CPUE. We see a positive association with Topeka shiner presence in restored Oxbows only. And a similar association with black bullhead, where Topeka shiner presence was positively associated with Topeka shiner presence in, or with black bullhead CPUE, sorry, in restored Oxbows. And we don't see any significant relationships for white sucker CPUE or species richness, so we'll skip to common shiner CPUE, where Topeka shiners are positively associated with common shiner CPUE and restored Oxbows. And we don't see any significant relationship with green sunfish CPUE either. But for dissolved oxygen, we see that Topeka shiner presence was positively associated with dissolved oxygen and unrestored Oxbows. So a quick summary, Topeka shiner presence displayed several relationships, positive relationships with some of the variables. So fathead minnow, brassy minnow, orange-spotted sunfish, black bullhead, common shiner, and dissolved oxygen all were positively associated with Topeka shiner presence, and most of those associations were seen in the restored Oxbows. And then to summarize what I said earlier, Topeka shiners were sampled more often in restored oxbows, and they were also more abundant in restored oxbows. So overall, the restored oxbows are showing positive results as far as Topeka shiner population goes. So that concludes my presentation. I don't know if we're doing questions right now or what's happening.
There's time for maybe one question. We're almost done. Okay, let's take Nick. We're getting there. We're almost back to the original schedule. I think we're just a couple minutes off at this point. So very good. Our next speaker will be Alex Bybel from Iowa State University. Alex is a MS graduate student in the Department of Natural Resource Ecology and Management. He has a BS in Fisheries Biology from Mansfield University of Pennsylvania, and he worked as a fisheries technician in Pennsylvania and Alaska prior to coming here to Iowa.
All right, so yep, my name's Alex. I'm going to talk about the genetics portion of this Topeka Shiner project we have going on at Iowa State University. Field work was kind of the same thing. Nick and me did the same research over 2 years and used the same data, so you'll see a lot of our methods are going to be the same. So kind of skip through a lot of the intro because Alicia and Nick already covered it, but some important points for my study I like to bring up is, so the Topeka Shiners, drastic reduction in their current range compared to historical. So in blue is the approximate historical range, and in green is a current distribution at the Huck 8 level. So you can see they're not near as abundant there as they used to be. Habitat loss generally due to stream channelization, which lead to populations becoming fragmented. Topekas are going to aggregate around those better habitat areas, which is why we're finding them in these off-channel habitats such as Oxbow with this slow-moving water. So previous genetic research done on the Topeka shiner, since it's a federally endangered species, there has been research done before. In 2000, Ann Michaels in Kansas did her PhD, and as part of it, she compared the Topeka shiner's mitochondrial DNA sequence across its range. On the right there is left is a picture of her study area. She found that there was 3 populations of Topeka shiners genetically distinct. So you have the Minnesota, South Dakota range, the Iowa and Missouri range, and then Kansas range. In 2008, Anderson and Sarver developed polymorphic microsatellite loci for the Topeka shiners, and they got this from Minnesota, Missouri, and South Dakota. These were used by Blanket All in 2011 in South Dakota. It was a project by the South Dakota Department of Transportation to look at the effects of culverts as barriers to genetic variation, or genetic gene flow in Topeka shiners, which was kind of inconclusive. What they did find was genetic variation between the 3 Huck 8 basins that they sampled. So what do we want to add to this study? So first of all, we think we can assess the genetic variation of Topeka shiners across the range from microsatellite loci. So again, get samples from across the range and compare the number of populations and the gene flow between them. So, and second, determine the habitat associations with the genetic diversity of Topeka shiners. And Nick's done a pretty good job looking at habitat associations with these oxbows and also in the streams from kind of just fish abundance perspective, but to add a little bit of layer to it, what does it do to genetic diversity? And then the third thing is to look at population structure in Topeka shiners, mainly in Iowa and Minnesota, because that's where the majority of the study was focused. So again, fish sampling, same as Nick's for the oxbows, we just seined them. Streams was a little bit different. We used one pass of electrofishing, then followed by as many seine pools as we could reasonably get in an area that was 20 times the wetted width of the stream. So, because there's sticks and rocks and everything, you're not able to get a seine through it very efficiently in some areas, so you do the best you can. So this is what our collection looked like. As you can see, the black dots there are the sample sites, mainly congregated around Iowa and Minnesota basins, so the Rock, the Boone, in the North Raccoon. That was kind of where the study was focused, and we were lucky enough to get samples from across the range from a couple of different peoples, the South Dakota Department, or South Dakota Game and Parks, as well as the National Park Service and U.S. Fish and Wildlife were able to help us out there to get samples. So at each site, there's 20 anal fin clips. If we could get them, it's a rare fish, so we didn't always get 20. Sometimes it's 15, 10. Or we could get preserved in 95% ethanol and then later extracted them using the Quiegen DNeasy blood and tissue kit. Overall, about 800 samples from 43 sites throughout the 2 years that we sampled. And then, so we did PCR on these 13 different loci. We were looking at 8 developed from the previous study I mentioned, and then another 5 that were polymorphic for Topeka shiners from other studies. The PCR was carried out in our lab by me and a bunch of technicians, and then genotyping was done by the Iowa State DNA facility, and they gave us back these nice little genotypes that you can see on the left. So this is where I'm at right now. I'm currently trying to call my alleles for these different loci. Across the top you can see the different basins, and across the side there are the different loci, and that's the number of alleles. We only had 9 loci show polymorphism, so 4 of them they'd have 1 allele at each thing, so it's just fixed, so you really can't do anything, so they're excluded from the study. This just kind of shows that, yeah, there's a lot of variation between those, and this analysis coming up will really kind of tell us the story of what's happening there. So again, the first objective, looking at this range-wide genetic comparison, so why you would want to do that. So these basins that Topeka shiners are currently located in are isolated by large rivers, and as a minnow species, they're not generally known to travel these large river distances. So we're assuming they're different populations, and these different populations could represent different management units. And then also too, we don't know the level of gene flow between them. Like, we're assuming that there's not much, but again, even within a subbasin, how much is there? That's what we'd like to find out. So how we would do that? So finding the number of populations, we have 8 different basins, so doing a simple structure analysis, it's a Bayesian clustering analysis, essentially giving you the number of populations based on the individual's genotypes, an AMOVA analysis of molecular variance, essentially telling you if those populations then are significant, and then assessing the variation between and within these populations, so doing that through a couple of different ways to kind of get the story. So FST, so it's a measure of genetic distances between populations. Things like allelic richness is the number of alleles corrected for sample size, heterozygosity, and then for within FIS, which is an inbreeding coefficient, which is going to be particularly interesting in these isolated populations, how much inbreeding is there. Objective 2, so this habitat association with genetic diversity. So again, using this, the influence of the environmental factors kind of to optimize this restoration success. Doing it from, this abundance side, but also looking at it from the genetics is, again, helping to give another layer to that. So we're going to use this program called GUESS. So essentially what it does is take genetic structure information, so F values, and compares them to different measures of habitat. So on the left we have just a bunch of different habitat we can use. Currently we're going to be working with Nick, kind of using his data in terms of what he found and significant habitat relationships with the Topeka shiners to kind of narrow down our list and kind of focus in on it. But again, it kind of, you put it in and it tells you what factors are going to be most important to genetic diversity. So this third kind of idea, this metapopulation structure. So what we don't know, the kind of the population dynamics of Topeka shiners, how far they're moving, how isolated these populations are, their gene flow between them. We do know that the populations are fluctuating with periods of drought and periods of flood, so they'll potentially either move or die during a period of drought because there's no water, so there's no fish there. And then in flood, we know these Oxbows flood and they're inundated with water, and also Topeka shiners, so they're moving around. And restoration is being carried out under the field of dreams hypothesis, so you put them in there, and yes, the Topeka shiners are going in those Oxbows, but how exactly are they utilizing them? So the way that these populations could be behaving in these areas. So a single pangmictic population will essentially means that everything's breeding, there's no restrictions to it, there's no non-random mating, there's no selection going on. So essentially the genetic variation will stay the same within this population. So isolated subpopulation, so it won't be in Hardy-Weinberg's essentially because they're separate populations, they're not mixing, so you'll see a deficit of heterozygotes loci will become fixed. And then there's this idea of an interconnected metapopulation. So population structure kind of violates your Hardy-Weinberg because you're going to see you're going to see differences in genetic variation because of this gene flow between the populations, but there's going to be kind of distinct populations within that. And then again, to kind of figure out how much between that, then we can look at estimates of genetic distance. Again, FST is known. We can look at nays and chord distances as well. So kind of the benefits to look at this. So again, looking at these genetically distinct populations as management units could benefit the species in specific managing. Habitat associations, again, the previous two presentations that, we're restoring these, trying to make them as best as we can for these Topeka shiners. And again, population structure could also help in the same way. How close do we need to make these? Are they able to travel far to go? Are these population mixing? And with that, I'm done. So.
So Courtney Zambory is our speaker up next from Iowa State University. She's an MS graduate student in the Department of Natural Resource Ecology and Management. She has a BA in Biology, Ecology, and Conservation from Boston University. And she worked as a fisheries biologist in Louisiana prior to coming here to Iowa. And her talk is titled Integrating LiDAR Landscape Analysis and Species Distribution Models to Prioritize Areas for Oxbow Restoration.
Okay, thank you guys for sticking around. I am the last person that's going to be speaking at this session, so after this we'll have a break. My project is a little bit different than the previous two studies we've talked about. I don't have a field season, so all my work's done behind a computer looking at GIS data and high-resolution satellite data. So we've talked today about off-channel habitats. These are bodies of water that are disconnected to the stream channel for most of the year during base flow. When there's flooding, they do reconnect and allow for movement of many species between the stream and this off-channel habitat. So they become increasingly important in conserving not only Topeka shiners that we've talked about today, but in many other aquatic species. These off-channel habitat restorations have been used in the Pacific Northwest in preserving coho salmon populations, as well as delisting the Oregon chub. It was the first endangered species to be delisted from being endangered, not because it went extinct, but because it was delisted to threatened. So the goals of this project is to use high-resolution LiDAR imagery to identify these areas of former stream meander in Iowa's highly modified landscape, and have those designated as potential restoration sites, and then integrate them with species distribution models for Topeka shiners specifically to identify where the best places to put these off-channel habitats would be. So the first question that I'm going to be talking about is, where can we restore? So Alicia talked about how critical it is that these restorations have groundwater connectivity throughout the year so that they are protected from complete desiccation. These fish cannot breathe air, so if the entire oxbow dries up, they die, and they're already endangered, so we try to avoid that. So this talk is looking and finding out where those streams used to meander, having those be our candidate sites, because those depressions are more highly connected to the groundwater table. You don't have to dig as deep, and it's always good to restore something that used to be there rather than just creating an entirely new area for restoration. So the way we do this, we use Iowa and Minnesota's statewide coverage of LiDAR data. So LiDAR data is high-resolution data. Each pixel on this image here represents a 2x2 meter square on the Earth's surface. We use that data to create a digital elevation model. So high-resolution elevation model, vertical resolution is in centimeters, so you can find out these depressions really easily. We limited our search for these potential oxbow sites within a 500-meter buffer of the stream channel as our riparian area. So at the bottom, you see that original digital elevation model. You can kind of see the Oxbows sticking out. Using GIS, we fill those depressions up, create a filled DEM, and then subtract the original from the filled to find those depressions, which is on the equal side, those areas that are outlined in black. So we have these depressions in the landscape, these riparian depressions. How do we start eliminating them to find those that are former stream meanders and oxbows? Well, first we eliminate any depressions that are less than 100 square meters. Those are considered too small to be viable restoration sites. So we get rid of those. By definition, an off-channel habitat is not connected towards the stream channel, so we eliminate all depressions intersecting that mainstream center line. And lastly, we took depressions that we eliminated depressions that had less than 100 square meters outside of an agricultural field. Alicia also talked about how these depressions are, or they do the restorations in pasture lands, so cattle moving through them is totally okay. It's actually really good because there aren't a lot of trees usually in those pasture lands, but of course we all live in Iowa and know that doing restorations in the middle of a farmer's cornfield is frowned upon, so we limit those depressions as well. So now we have a pool of potential oxbow sites to restore. So each of these depressions has multiple characteristics calculated for them using GIS data. So first of all, we look at morphometric characteristics. So we look at circularity, which is defining how similar that shape is to a circle. Solidity is a characteristic defining how convex and concave shape is. Rectangularity, how similar to a rectangle. And then individual convexivity and concavity metrics. And additionally, we look at the topographical characteristics of those depressions. We look at mean and maximum depth, mean slope of the depression, and mean roughness. And lastly, we look at mean height above channel. So that's what you can see depicted there on the right. That's a mean height above channel map that we created using the USDA's Agricultural Conservation and Planning Framework tool. So the way we calculate that is in the middle of that stream that you see in the blue, you've got a white burned mark set as your 0 mark, and then each pixel that flows into that stream is calculated how high it is in centimeters above that 0 mark. So right off the bat, you can see those areas where oxbows used to be pretty clearly, those blue sinuous areas that are disconnected from the stream channel. Blue is closer to the same elevation as the stream itself, and then you get warmer as you move higher and higher above that riparian zone. And those white, gray areas are more than 3 meters above the riparian zone itself. So to develop this model, we take all of these depressions, and this was a little intense, but we went through and coded all of them as either a target feature, so a oxbow or former oxbow feature or stream meander that we identified using many years of historical imagery back to the 1930s, or a non-target feature, so a pond, farm to pond, a field depression, or such. So those areas that we weren't looking for are hatched out, and you can see those oxbows. The left image is aerial imagery from 1930s, and the image on the right is from 2015. So you're not meant to read this, but this is just to illustrate that we created models for each watershed of study, the Boone, North Raccoon, and Rock River watersheds. We also created a model just using those two watersheds located in the Des Moines lobe, which are the Boone and North Raccoon. And finally, we lumped all the watersheds together and all the depressions within all those watersheds to create an all-watershed model. We ran these models with the classification and regression analysis, and we noticed that shape factors like solidity and convexivity or concavity were the first split in all models. So that shape is really important in identifying these former stream meanders, which makes sense. If you think about an oxbow, they are horseshoe shaped, or a stream meander is going to be very curvy. The next split was always mean height above channel. So that was, as predicted, a very important factor. Once these oxbows cut off from the mainstream, they do remain that same elevation, relative elevation to the stream channel until many years later when they silted in. I think Alicia said after 17 years they only had 3 inches of siltation noticed in many of these places, so you can identify those really well. This model is the watershed model. Those nodes that are characterized as gray ones and ones were nodes that selected for these target features, so for former stream meanders and oxbows. And the numbers below them is conditional probability of each node, so how much noise, or 1 minus how much noise was associated with each terminal node. So higher numbers mean that when tested against our test site, we had a lot less noise associated with those areas classified as target features. So our results, overall our models performed really well. We evaluated them using the area under the receiver operator curve. So it's kind of on a grading scale with 0 or 0.5 being a failed model. It doesn't perform any better than random. And as you get closer to 1, you get a better model. So all of our models, each individual watershed as well as all watersheds together, performed in the B to A range. So we're pretty happy with that. Our data using all combined watershed data performed just as well or equally as well to the other watersheds, so we decided to use that. We did have a considerable amount of noise. Any error associated with those models does reflect a little bit of error, so we ranked those models that were calculated as Oxbows from 0 to 7 based on those conditional probabilities. So the higher the conditional probability, the higher or the more likely it's to be a true Oxbow or candidate restoration site. So we'll move quickly into where should we restore. So we have these Oxbows, these potential candidate sites where we want to restore, but based on where Topekas are, how do we select them? So we used data collected from 2010 to 2017 to generate species distribution models for Topeka shiners. We used models from both habitat types, but I'll talk about Oxbows for this session. We used the species distribution modeling package Biomod 2 in R and developed many single model approaches and a total ensemble model approach, which I can talk about in detail to anyone who's interested. But here are the results for each individual watershed. Again, we evaluated the AUC score. We had varying individual model results, but then we used a weighted average to create that ensemble model and applied those results to our pool of potential restoration sites and ranked each site based on their suitability, looking at just landscape associations with those Topeka shiner distributions. So for our ensemble model, this AUC score has performed really well, so 0.8 to 1, so almost perfectly modeling, just based on a testing data set that we looked at, where those Topeka shiners could be. Now, further sampling is needed to evaluate this metric, but we kind of have a starting place of where we can start targeting restoration. So in conclusion, as we talked about, conservation of highly modified landscapes presents a suite of challenges of finding where you can restore. We found that using this LiDAR data, we can successfully extract these features from the landscape, and then combining those features with species distribution models, we can start to kind of target areas that are suitable for restoration for this. And then because Oxbow restorations are critical for a lot of other species, we think we can apply this in other regions and for other different species and habitats. So thank you. Any questions?
We have time for a question.
Yes.
In the context of the modeling I'm identifying, did you see any benefits or any places that correlate better to the precision within the stream and the watershed, farther upstream versus downstream?
So in our models, when we looked at Oxbows, we didn't see any really important factors with stream order. However, we did see a factor in percent water within different catchments, so which could be correlated to how big that stream is. And so these species typically like smaller headwater streams, so between 1 and 3. So yes, restorations should be performed more on those tributaries rather than the North Raccoon River watershed itself. Those are a little big for the species.
Any other questions? Great, well, let's thank Courtney. And now for that all-important break, we're a few